Hello, welcome, and thank you for joining us for today's webinar, Wireless Optogenetic Stimulation for Rodents. My name is Charlene Herndon, and I am the Global Product Marketing Manager with Harvard Bioscience. Today's presentation will explore wireless optogenetic stimulation, the features and benefits, as well as important surgical techniques for this application. Before we begin, I'd like to address a couple of housekeeping items for the webinar. First, this webinar is being recorded, and after the event, everyone will receive a link to the recording via email. We'd like you to sit back and enjoy the webinar today. Don't worry about taking notes, as you will be able to view the presentation again at your leisure. Second, if you'd like to ask a question during the event, feel free to type your questions into the chat box located at the bottom right-hand side of your screen. These questions will only be seen by myself and today's speakers. At the end of the presentation, I will read the questions aloud for the speakers to address. Now that we have reviewed these points, let's move on. We will have two featured speakers with us today. The first will be Dr. James Maurizio, General Manager for Triangle Biosystems International and Director of Research and Development with Harvard Apparatus. Our second featured speaker will be Mark Rossi, a National Science Foundation Research Fellow in the laboratory of Dr. Henry Yin with the Department of Psychology and Neuroscience at Duke University. And now I'd like to turn the floor over to Dr. Maurizio.
Thank you, Charlene, and welcome everybody. As Charlene mentioned, we will be discussing the wireless optogenetic stimulation system that Dr. Yin's team and Mark Rossi will present after my presentation. The overview of my presentation will include evolution of optogenetics and how we evolved to the system we have today. I will go into some of the key features of the TBSI wireless optogenetic system, discuss a little bit about the stimware and software interface, show a little video on the actual OptoStim head stage working with a zoomed-in camera shot, and then I'll turn it over to Mark for the stereotactic and surgery and experiments that they used at Duke University using this equipment. I want to mention a few classic publications on optogenetics. These were written in research back in 2005 with Dr. Dezeroff and Ed Boyden's team. The technologies for these approaches and techniques have evolved over the last decade, and the motivation for this work and the design that you'll see today was really for freely moving animals, which is the space that we at TBSI are kind of interested in pursuing. So we have, as you know, wireless recording and wireless stimulation, and this wireless optogenetic stimulation system that I'm about to present, it will fall in line with that market size. Okay, and so let's discuss the optogenetic system. Traditionally, we had a light source here that my pointer seems to be not working. Ed, could you? Thank you. The classical optogenetics stimulation systems include a strong light-powered source. It could be an LED or a laser here, controlled by either a function generator or a personal computer. At the end of that light source, there's an optical lens focusing the light energy into a fiber optic cable, which goes through a commutator, and note it's an optical commutator. And the light continues down a path of after the commutator to another fiber optic cable to what we call an optical ferrule that converts the fiber optic connection from the commutator into the implanted fiber that is at the actual anatomical target of the brain, spinal cord, or peripheral nerve that you're interested in stimulating. And there's where there's the channel rhodopsins in the targeted area for inhibiting nerve stimulation, or the halorhodopsins to inhibit the nerve stimulation, or the channel rhodopsins to stimulate the nerves. The evolution is taking place where we have the light sources moving from before the commutator to after the commutator. These are systems that have come out in the last couple of years, Plexon has a system similar to this, and some other companies, where you actually put the light sources after the commutator. This allows for electrical commutation, which is a lot easier, and it will also allow for multiple channels. You know, this system up here, it's difficult to do more than 2 to 3 to 5 channels of optical stimulation through a commutator. Electrical commutation is a lot easier, and you could do many more channels through the commutator. And so if you can move the light source after the commutator, you can take advantage of the electrical signals to do that. And you still have the same lens, some fiber optic connections. The ferrule's still there as well, and the fiber optic, the fibers, you know, into the brain area are still the same, with the same techniques as above. So what we've evolved in the last 2 years is what we call an optrode, or an implanted optrode light source. We'll talk more about that later on in the presentation. But we have a stimulation system here. Again, my pointer doesn't seem to be coming here. Ed, could you give me the pointer, please? Oh, it's down here. Okay. So what we have is our same setup as the previous slide. We have the electrical connections coming from a function generator or a computer. It goes through electrical wiring, but we have a very big distinct difference here, and that's this electrical connection through the skin barrier to electrical wires feeding the light source, and there's no lens here, and there's no fiber here. So this is the light source directly at the implanted site using our optrodes. We have a variety of different optrodes that we'll talk about later. And the key advantage here is we can come up with many wires into the animal's brain, and we can theoretically do up to 16 or 20 or 32 light sources or LED sources at the tips of these optrodes. So we basically expanded the channel count for optogenetics manyfold in doing this. The second thing is the optical efficiency of this approach is a lot better. Every time you actually go through a fiber connection and an interface, you lose optical efficiency. Even the lenses are actually suck up a lot of optical power as well. So the amount of power and energy needed to do an optostim like using this approach with these optrodes is a lot more efficient when you compare the electrical power you use to the optical power you receive. Converting this over to wireless is pretty straightforward in the sense that you just break the electrical commutation and the wiring, and you go with our USB digital radio to connect the PC to an electrical stim head stage, which we call our OptoStim head stage. We have the same electrical connections going into the brain, but we have the same electrical wires and optrodes feeding it. So we just basically removed the electrical commutator and the electrical wires with our wireless connection here. So in review, we have our stimulation system that includes the OptoStim software running on a PC. We have this wireless USB connecting dongle that we can connect to the head stage of the animal that's mechanically mounted, and then we have the optrodes that are implanted in the brain. This is the top view and side view picture of the head stage. These sizes can vary based on, and the weights of these can vary based on the size of the battery. We have many battery options, and so the weight and size can be tuned for the animal species or adjusted. This is a 2-hour battery that weighs 2.8 grams, which is similar to what Mark Rossi will be presenting later. I want to point out a couple important things, and that is these trigger connections here on the USB dongle. You can't really see them there, but there are 2 TTL trigger inputs that we can connect to the cage or the operant conditioning cages that you want to run your animals in, and so we can trigger these the stim patterns once they're downloaded into the head stage, and you can actually get very good resolution on trigger delays, and which will allow users to get sub 10 millisecond trigger delays with 100 microsecond accuracies on triggering. I must also point out these are the latest vintage of optrodes. We're running around 550 microns wide, and that's the width of that flexible shank. We also have different depths. These are the 4 millimeter depth there, and these are the 8 millimeter depths here. These Optrodes have two LEDs positioned. You can't see them, but there's two LEDs positioned very close to the tip of that shank. And then the wire, the Omnetics connectors here come out, or we can come up with a variety of connectors that connect into our Optrode flex cable. The key features of our system include, well, we have the ability to do two independent stimulation channels with different pulse current and pulse amplitude for the LED brightness levels. We have 12 bits of accuracy for the current, so you have dimming levels of quite a lot to brightness levels. We also have many different accuracies of timing and pulse widths as well for the stim pattern. We'll talk more about that later. Our current LED is a blue LED with wavelength 465.6 nanometers. This is from a company local in the Triangle called Cree, and we use that on these Optrodes to put and to give us the optical energy we need. The 3-hour battery is one standard option, but we could change that, as I previously mentioned, and that is it will last. It's a 60 milliamp hour battery that we can run in this head stage as well. I want to mention our accessory, which is our inductive powering, which allows for 27 recording. So if you run months of optical stim or many hours of stim, you don't have to worry about the battery size and the battery weight. We can use inductive powering on that, and the weight differences are the same between typically an inductive powered accessory head stage versus a normal head stage. I also want to mention you can move the head stage off the head of the animals onto the back with a harness, and then have the cables coming off the head stage to the Optrode connector as well to lighten up the head on the animal for those customers using smaller rat pups or mice pups. This technology also works with our wireless recording head stages, so we can completely have both combo stim and recording going on in parallel on the animal. Next is the key features continued. I want to talk about the trigger delays. You know, currently if we could trigger from the dongle, we get 10 milliseconds of delay, which is pretty fast from the trigger pulse, and that is pretty much the fastest we can do. The variations of trigger delays go 10 microseconds, and that is very consistent amongst the window of experiments we've seen. We also have a custom Optrode lengths as well, ranging from 4 millimeters all the way up to 25 millimeters. I want to point out some key important attributes of these Optrodes in our system, and that is we can get, this is a graph showing optical power and percentage of current, which is the obviously the current feeding the LED itself. This is optical power measured off of a photodiode that we were able to calibrate using some expensive optical equipment. And so we got obviously a good linearity of optical power versus percent of current, which is the amount of current feeding the LED. So when you're at 100% current, that's 20 or that's 30 milliamps of current, and that could give us 10 to 12 or even 14, up to 14 milliwatts of optical power. Typical power requirements for Optogenetics is 10 milliwatts, so we're well above that. The other important attribute is the temperature change that we have to be careful of. You know, we did not want a degree C temperature variation at the electrode site, at the Optrode site, so we're very cognizant of that, and we can change the duty cycle with our software and change the peak current, which obviously has an effect of temperature. So as we bump up duty cycle and peak current, obviously the temperature near the Optrode will go up. And you can see if we want to maximize a 1 degree option for degree Celsius temperature increase, even with the 25% current of peak current, we're excuse me, we're still below the 1 degree temperature change, and so we have the ability to monitor that, and we calibrated that with our equipment. I want to also point out these web links for more detailed descriptions of what you see here in our brochure and the manuals. So I'm going to show right now a quick short video of the OptoStim head stage driving LEDs. This is an older vintage of our OptoStim head stage and Optrode. This is the Optrode connected to the bottom of the head stage right here. You'll see one LED at the tip and one LED, I think it's like 1 millimeter up the shaft, which you'll see here, both flashing. This is at the bottom of the head stage that gets mechanically mounted on top to this connector interface here. By the way, this is a charging connector over here, and this is our programming, a microcontroller programming port here that you see here. This is our antenna feeding to the dongle. So why don't we begin the video here, and you'll see the actual LEDs firing at different rates. So the bottom and top are both firing, and you'll see similar brightness levels. We could do different brightness levels here as well with different pulse timings. The top LED you noticed was firing at at a smaller or a lower frequency as the bottom one, and so you can control, again, this demonstrates the ability to fire different timings and different duty cycles for each of those LEDs. Thank you. So moving along, I know it's difficult to read this slide. This is a little interface for the programming the actual excuse me, the actual channel's timing. You'll see here a pulse amplitude and a pulse width of the single pulse that we use for the each channel, and that pulse can get replicated in what we call a pulse train with some frequency that's defined here, and then that train can get replicated with another set of frequency and controls here. So you can do very, very long stimulation patterns over several days, and you could define a simple pulse and replicate that and do some creative patterns if you like. This is the channels here, and for channel 1 programming you fill in this table. It's simple. For channel 2 programming you do the same table, and I want to mention this search head stage button is the way to, we could control up to 16 OptoStim head stages with our system individually, and for grouped house animals or a rack of cages, so that one dongle can program by selecting the appropriate head stage here these different patterns. And you could trigger the pattern after you download it through the software interface, and I mentioned those trigger delays are more like 250 milliseconds to 300 milliseconds with quite a bunch of variability in trigger timing. It's probably almost 100 milliseconds of variability. For some users that's okay, but mostly it's not for very critical timing experiments that care about behavior and the timing with all that interleaded with that. So with triggering the software through the dongle, it's basically a lot more accurate, as we said, and we could talk more about the software moving forward with future webinars. But I just wanted to introduce the menus and how we actually interface and control the head stages. Okay, so at this point I'm going to turn it over to Rossi as the presenter, and Mark, take it away.
Hi, thanks, Jim. So my name is Mark Rossi, and I work in the lab of Henry Yin at Duke University. And today I'm going to be talking about some of the experiments that I conducted using the TBSI wireless optogenetics system. So optogenetics allows researchers to use light to activate discrete populations of neurons with extremely high temporal and spatial specificity. And it's proved a powerful tool for researchers recently. But the problem with traditional optogenetics is that it requires a mouse to be connected to an external light source. So this is usually accomplished with fiber optic cables, as you see in this picture. So these cables run from the mouse's head to a laser, and it allows the light to be delivered into the brain of the animal. But being physically connected to the laser constrains natural movements. It can greatly restrict the distance that animals move from the light source, and it can also introduce torque to the cranial implant, and it can perturb free movement. This also limits the number of freely behaving animals that can be tested simultaneously. So for example, if you have two mice that are freely interacting and they're both connected to a laser via these cables, then they're going to inevitably become tangled. So to begin an experiment, the first thing that you need to do is to implant the Optrode. So this is accomplished using methodologies similar to any standard intracranial implantation. So the first step is to anesthetize and head fix the animal within a stereotaxic frame. So the stereotaxic frame looks like this, and the animal's head gets held in place between the ear bars. So this is just showing it from another angle. And then a craniotomy is made over the target location, and then you slowly lower the LED shank into place and secure it with dental cement. So the LED shank you see here, and this is being held with a custom electrode holder that we made in our lab, but you could also use standard stereotaxic adapters to hold the Optrode in place while you're doing surgery. So this is an example of a mouse after the surgery has been completed. So you see here that the implant is fixed in place on the mouse's skull, and this is when the head stage is connected. And so just as a note, this is one of the very first designs for the Optrodes, and so it tends to be a little bit longer than the ones that are currently available. So this particular example is protruding a little farther off the head than what would normally be expected with the current designs. And so for the experiments that I'll be talking about today, we used transgenic mice that express channel rhodopsin 2 selectively in neurons that express the D1 dopamine receptor. So this population of neurons, also known as the striatonigral neurons, are known to be involved in the regulation of movement. And so we used this particular type of mice because we know that these neurons can control movement, so we thought it would be a good way to validate that the system is functioning properly. And what you see here is an example of histology from a mouse following the completion of experiments. And so the LED location is indicated here by the arrow. And so this is a pretty typical profile of the damage caused by the Optrode, and it's pretty comparable to most cannula implantations. So in order to test the involvement of the striatonigral neurons in movement and to validate that the system is working, we first stimulated at high power, so this is 100% power. And so what you're going to see is that this is a freely behaving mouse who has a unilaterally implanted LED Optrode. And when the LED comes on, you see that the mouse begins convulsing and twitching. So these twitches are very robust, and it was consistent across all mice that we tested. So when the stimulation ceases, the animal will quickly go back to normal behavior. And it should be noted that while the weight on the mouse's head doesn't seem to really interfere with any sort of free movement, over time the mice become adjusted to the weight, and they actually strengthen their muscles and no longer have any problems bearing the weight on their head. And so as soon as the laser goes off, the mouse stops twitching. So now the LED is off, and the mouse goes back to behaving freely. So this is what the quantification looks like of those data. So as I said, the behavioral response was consistent for all mice. So we calculated the rate of twitching as a function of pulse frequency. So the video you just saw was the high frequency condition, the 20 hertz stimulation. And we see that the rate of twitching increases as a function of the pulse frequency. And during baseline conditions in which there's no stimulation, the twitching is not present. So to test the long-term stability of these LEDs, we ran the same set of experiments 41 days after the initial test. So the video you just saw was from the initial test. And 41 days later, the behavioral repertoire is preserved. So we see this nice linearity with increasing twitch rate as a function of pulse frequency. So this is more than 50 days after the implantation surgery. So these LEDs are quite stable over time. So it's suitable for chronic experiments. And because we used very high power at about 32 milliwatts for the experiments that I just described, we wanted to make sure that these results were not due to non-specific heating of the tissue, which can accompany this type of stimulation. So we did the same experiments with control mice. So these are D1 mice, D1 Cree mice that don't express channel rhodopsin, and we see that there's no effective stimulation. So twitching is not induced at all by the stimulation. And so we saw these robust convulsions during the high power stimulation, but previous reports had shown that if you stimulate the striatonigral neuron population unilaterally, that you can produce controversive turning, that is, turning away from the side of stimulation. So what we wanted to do was attempt to verify that result. And so in order to do that, we reduced the power of the LED stimulation, and we recorded during free behavior. So this is an example of a D1 channel rhodopsin mouse that has the optrode placed in the right hemisphere of its brain, in the striatum. And when the LED comes on, you see this robust circling behavior. So he's circling to the left. That's controversive to the site of stimulation. And with this type of stimulation, there was a slight lag in the offset of the behavioral response. So for a couple of seconds after the laser or the LED went off, we see that the animal still prefers turning in the controversive direction. So the LED is off now. And then that quickly rebounds, and the mouse can move quite freely right after that. And again, you see that the overall locomotion of the animal is not impeded by the weight of the head stage. So to quantify that, we have this here. So this is just showing a schematic of the site of stimulation in the right hemisphere produces turning, the controversive turning, so turning toward the left. And if we quantify the time, the proportion of time that is spent turning during baseline conditions, the black lines here, we see that very little time is spent turning. The mice tend to walk in straight lines or not walk at all. But when stimulation is present, there's a very large increase in the proportion of time spent turning, and it tends to be heavily biased toward the controversive direction. So they're turning away from the site of stimulation. And this is in agreement with previous published reports. So it verifies that this stimulation is producing similar outcomes to other optogenetic stimulation experiments using traditional fibers. So in conclusion, the wireless optogenetic stimulation is suitable for use in freely behaving mice. And high-power stimulation of striatonigral neurons produces frequency-dependent twitching. And low-power stimulation of these same neurons produces controversive turning, which is in agreement with previous reports. So I'd just like to thank everybody for their time And now I will hand this back off to Dr. Maurizio, who's going to talk a little bit more about the wireless optogenetics setup.
Thank you, Mark. I wanted to thank also thank everybody for attending the webinar. I wanted to mention that we are going to be doing future webinar topics in optogenetics. If there's an interest, there will be some more discussion on stimware training and how to use it and control these wireless head stages, either in a grouped house situation or a singular situation. I'd like to eventually brainstorm about future directions that we see with the wireless connections. Obviously, we want multicolor LEDs and smaller head stages in the future. That's all coming down with more channels as well. There's a lot of discussions about how to do bilateral optostim as well with our technologies, and a lot more optrode options combining the recording and stimulation on the same shank as well. So we're only going to continue to improve what we have and carry forward with R&D, more technologies there, and getting your opinion on that to help us steer us in the right direction is important. And then, of course, application areas, brain, central nerve, peripheral nerve. There's with freely moving animals, you can do a lot with combo sim and recording in this space, and we like to kind of get focused on what's critical in the field and where people are going with this. At this point, I'm going to turn it over to Charlene and for a question and answer period.
Okay, thank you. I'd like to thank Dr. Maurizio and Mark for a wonderful presentation. And as Dr. Maurizio mentioned, let's turn it over to question and answer. And let's see. Our first question was, do you have options for remote triggering from other systems, say operant or video tracking?
Yes, we do. As I mentioned previously, we have those trigger connections coming in. They're TTL pulses from the cages that we can directly trigger the USB dongle with, and we can get very good trigger delays through that within 10 milliseconds with 100 microsecond accuracies. We also have the ability to interface to different software packages. This OptoStim can read a file on a computer that another neural recording system can generate. So we can continually monitor a file and trigger stim patterns and even adapt stim patterns based on this file read and write handshake. Now I want to mention though that every time we go do that triggering, it's a lot more delayed, but it is doable, and you'll go from a 10 millisecond trigger delay up to a couple hundred milliseconds through the USB dongle. So yes, that's possible, and we've like I say, have solved that problem.
Okay, thank you. The next question is, what is the depth of the optrodes for spinal cords? And are they usable for adult mice?
Okay. The optrodes we offer, and what I've seen in the field so far with people stimulating spinal cords, are they lay the optrode parallel and flat onto the spine, and the LEDs face down into the spine tissue. So implanting the spine or implanting an optrode in the spinal column, I'm not really sure how they do it and what depth they have for that. Mark, maybe you have an opinion on that as well. But all the spinal cord stimulation I've been aware of is they're just laying it on the surface, and they're not really penetrating the spine. The light's so bright it will shine through a lot of tissue. So I don't think it's an issue. But I think there's these optrodes you could bend 90 degrees. So you can theoretically turn them and bend the metal flex shank down 90, and with tweezers do some insertions if needed, or some type of stereo tack. Mark, do you have an opinion on that?
All of the experiments that I've done have been in the brain, so I've never done spinal cord implants, but presumably if you laid it across the spinal cord, the light would be sufficient to activate neurons within the spine.
Okay, thanks.
Okay, the next question is, for delays of 10 to 250 milliseconds, could the delay of a specific stimulus be known or recorded?
Well, the delays, well, we actually did that in calibrating our trigger delays. We actually had a photo diode monitoring when the diode turns on, and then we trigger the stim pattern either with the trigger ports on the USB dongle or through software. So we actually have done that in vitro, by the way, outside the body, so to calibrate the stim delays and stim accuracies. We also could monitor and record while stimulating, and as long as the timestamps of the stimulation system are synced to the timestamps of the recording system, you could actually see that as well. And so, yes, the delays that we talked about were accurately monitored, and so yes, they can be, and the data that's in our specs and manuals reflect that information. Thank you.
Okay, next question is, what is the substrate of the Optrode? Is it rigid or flexible?
Okay, the actual material is a polyimide with gold top and bottom routing and metal layers. I think the gold thickness is 22 micrometers, and the flex material, the polyimide, is I want to say 20 to 50 microns thick. So that polyimide is flexible. You could think of it as like a piece of tin foil, but a lot more rigid. You can actually bend it and it will stay in shape in how you bend it. You could bend it back and forth several times without breaking it, but if you go to a 90-degree bend, you're really stretching the gold wiring, and it will, you know, the reliability will drop off fairly quick. The flexibility of the Optrode is such where you can definitely insert it to kind of to different materials, brain, muscle, or whatever. So it's not going to bend and move in place when you do that. It's stiff and it's rigid enough, but yet it's not rigid where it's going to crack or break if it accidentally gets hit. So that's why we chose that material over some other stuff. Now I'm going to mention that, you know, there's a lot of new technologies coming out for Optrodes and that are going to be smaller and stuff. So, but yeah, this material is somewhat flexible, but it's stiff enough to do surgery. Mark, do you have an opinion on that?
Yeah, the shank is actually, the stiffness is very beneficial for surgery. So for example, I commonly implant glass fibers for chronic stimulation, and with those the surgery tends to be a little more difficult because if you bump it or if it happens to hit the skull while you're implanting, then the fiber breaks and you have to start over. But with the Optrode shank, it's rigid enough so that if it bumps something, nothing really happens, and if you can implant it, and it's rigid enough to go easily into the brain with no resistance.
Okay, thank you. Yes, this is what customers are doing now, and more recently in our database. What's happening is customers want to stimulate in different parts of the brain, bilateral stimulation or quadrilateral stimulation, and so what we have is envision the connector that mates to our head stage on the bottom is what we call an electrode interface board. So the head stage is physically mounted and removed on and off onto this electrode interface board, and then from the electrode interface board we have fly wires coming out of that, 36 gauge tiny wires that feed into the Optrodes that are the Optrode connectors that are in different parts of the brain. So the head stage is physically set, and then the cables will allow the users and the customers to implant at different parts without worrying about mechanical and stringent effects of, you know, how to space everything. So yes, that's a very good technique being done right now, and it's available today.
Okay, next question is, in the Rossi et al. 2015 paper, the battery was recharged periodically. How was this done with the battery? Was the battery encapsulated in epoxy? Was it done by induction?
For those experiments, the battery was encapsulated in the epoxy, and so it was not inductive powered. And before the experiments, I fully charged the battery, and then I was able to test multiple mice in the same session in series, and there was no power loss as far as I could tell. So it lasted comfortably over 2 hours.
Yeah, and I want to mention with inductive powering, we have obviously it's relatively the same weight as the head stage mark used in the picture there, and battery limitations are kind of muted when using that. The other thing is for very tiny animals, we can remove the head stage and put it on a backpack with a nice little silicone harness we offer. Thank you.
Okay, the next question is, what is the youngest age that mice can have for carrying the Optrode and head stage?
Yeah, so clearly we care about the weight on the head of the mice as well as the mice pups. I mean, that's been a big concern for all our wireless products if we want to go after the mice market. Clearly the rat market can carry a lot more weight. I mean, we typically target 2.8 grams for an adult mouse. I don't know what and the age curve and the weight curve of a mouse pup can carry, but I want to mention, I kind of alluded this to the last question, is that we can remove the weight, the complete weight of the head stage, and actually move it to the back with this kind of very elegant silicone harness. And so the weight is actually removed from the head, and all that comes out of the harness is this little cable with a mating connector to the Optrode or Optrodes. So the weight of the head is literally 0.2 grams or 0.3 grams, very tiny, and but the pup is now carrying a harness, which is it's 4 silicone tubes that wrap around it in a nice, elegant way, and it keeps the animal as comfortable as possible without having all the head stage weight. Mark, do you have an opinion on the weight and what you did and the strength of the muscles and stuff?
Yeah, so for the experiments where you're going to have the head stage actually on the mouse's head, I would recommend probably using adult mice, so not younger than about 6 weeks of age. And for these mice, they can tolerate the weight very well. With no training at all, it doesn't impede movement. If you do very long sessions, eventually their heads will, or their muscles supporting the head will fatigue. But we see that the mice are able to strengthen these muscles over time, and they become quickly adapted to having that extra weight on their head. So it doesn't really seem to pose much of a problem for an adult mouse.
Okay, the next question is a 3-part question. First, can the electrode be moved after implantation? And then will yellow LEDs be available? And can Optrodes be implanted bilaterally?
Yeah, so the Optrode, it presently can't be moved in the Z plane after implantation, but it could, in theory, be reused following removal from the animal. So I tested the Optrodes following removal, and they were still functional. So they came out of the animals still intact and straight, and could be implanted again if you remove the epoxy from around them. And I'd just like to point out that although presently there aren't yellow LEDs available, that you can still achieve optical inhibition using the blue wavelength light. So using the present configuration, you could there are options that respond to blue light that can be used to directly inhibit neuronal populations. So that's certainly an option even before the yellow LEDs are available.
Okay, thank you. And now, to be respectful for time, I think we'll have one last question. The last question will be, how close do you need to be to the target neuronal population to record?
So this is pretty variable, but presently there's not a simultaneous recording and stimulation option on the single Optrodes, but I believe that that's something that Jim is working on for future versions of this.
Right. Yeah, basically there's no reason why you can't put a recording electrode very close to the Optrode and pick up neural signals while stimulating or before and after stimulation. So there are, obviously we want to include Optrodes with both recording sites and LEDs in the future. And what I want to mention about, real briefly, about the artifacts of using these Optrodes for recording. Typical electrical stimulations, pump current in and out of a tissue sample, and you get major artifacts of perturbations in the nerves and the tissue until the charge balance goes away, and there's could be several milliseconds of delay. There are some artifacts or electrical artifacts of this type of recording, but they're very predictable because they're on the edges of the LED pump currents. So when you see a rising edge and a falling edge of the LED, you will see a little artifact in the recording piece. It's very predictable, and we could subtract that out in software. As compared to electrical stim, where you have full current going in, and what happens is typically the amplifiers rail unless they're switched on and off like some people do, and so we don't have that issue at all with this. So thank you. Good question.
Okay, and that concludes the question and answer portion. I'd like to take the opportunity to thank you all for attending our webinar today. We know how valuable your time is, and we greatly appreciate you sharing the better part of this hour with us. If we've missed answering any of your questions, I apologize, and we will be glad to follow up via email. I'd like to invite all our attendees to click on the link on this slide to download our recent publication from the February issue of Frontiers in Integrative Neuroscience. If you have any questions on the system or which configuration would be best for your application, please send your inquiries to sales@triangleBiosystems.com, and your local sales and application specialist would be happy to assist you. Thank you again for your time, and have a wonderful rest of your day.